Welcome back to botany class. In the previous class, we discussed a detailed account on gene mutation or point mutation. Today, let's talk about chromosomal mutation or chromosomal aberration. Define mutation. Mutation is a sudden change within a chromosome. What's the difference between gene mutation and chromosomal mutation? Gene mutation takes place or variations takes place within a particular gene. Chromosomal mutation, it's a large scale variations observed within the chromosomes. More genes, variations find in more genes. This is individual gene. Mutation takes place within a single gene or individual gene is called gene mutation. Large scale variations observed are called chromosomal mutation. Chromosomal mutations again classified into two different types because these variations are observed either in number of chromosome or in the structure. If the variation observed in number are called ploidy and another variation observed in the structure. Let's talk about this ploidy. That means the chromosomal mutation or chromosomal aberration occurs and by altering the number of chromosomes. You all know there is a basic set of chromosomes. There is a definite set of chromosomes within an organism. All the organism possesses a fixed number of chromosomes. It cannot be altered or changed. If any alterations observed, that's called ploidy. So this ploidy can be in chromosomal mutation. Chromosomal mutation can be observed or detected by microscopic examination or genetic analysis. Whereas in contrast, this gene mutation cannot be detected by this microscopic. Chromosomal mutations can be detected microscopically. Ploidy can be classified into two different types, aneuploidy and euploidy. Aneuploidy and euploidy. What's the difference among these two? Aneuploidy is the alteration of chromosomes within a diploid set. The diploid set we can say as 2n. Euploidy within an entire set of organisms. Alterations take place within the entire set of organism is called as euploidy. Aneuploidy, the number variations or alterations observed within the diploid set. This is the normal diploid set of chromosome 2n. Diploid set, normal chromosome number. If any alterations observed in this, either by addition or by deletion, if the chromosome number, individual chromosomes within the basic set is added or deleted or eliminated is called this ploidy. So based on this, it's classified into aneuploidy and euploidy. What's aneuploidy? Aneuploidy is the changes or alterations within the diploid set of chromosomes. In this diploid set, if any alterations observed are called aneuploidy. Within the entire set of chromosome, if any alterations observed are called euploidy. Aneuploidy is again classified into two different types, hypoploidy and hyperploidy. Hyperploidy and hypoploidy. What's the difference among these two? Hyper and hypo. Hyper by addition of chromosome, hyperploidy. Hypo, deletion of chromosome. Within this diploid set, if any number added in the normal set, if any numbers, extra chromosomes are added, that is called hyperploidy. If any number deleted is called hypoploidy. So, based on this, it's again classified into hyperploidy. Ploidy is again classified into trisomy, tetrasomy and pentasomy. Trisomy, tetrasomy, pentasomy. You know that tri, 3 in number, tetra, 4 in number, penta, 5 in number. How it's the alteration in number. Within a normal set, within the diploid set, the diploid set is 2n. Trisomy, if any individual set is added, 2n plus 1. So, it becomes 3, trisomy. So, within the normal set, this is the normal set. Here, trisomy, 1, 2, within this normal set, 1 is added. So, this is the extra. So, this is what? Trisomy. Sometimes double trisomy. Double trisomy means 2n plus 1 plus 1. One more. One more is added. This is the normal. Normal should not be changed. But if any changes takes place, that's called mutation. So, this mutation is ploidy based on the number. Changes in the number or variations take place within the number of chromosomes. So, it's a trisomy. The condition is trisomy. Then tetrasomy. Within a chromosome diploid set, 2 sets of chromosomes are added. So, it's called 2n plus 2. This is the normal set. In this normal set, this is normal, 2 are added, 2n plus 2. Then pentasomy, 3 are added. This is the normal set. Here, these chromosomes are added. 3 sets of chromosomes added are called pentasomy. 2n plus 2. Here, this is double trisomy, 2n plus 2. This condition is tetrasomy. 2n plus 2. This 2 added. Here, this one, pentasomy, 3 sets. Here 2 sets, here 3 sets. Here plus 1, 2n plus 1 plus 1. So, the condition is 2n plus 1, this condition 2n plus 1. This is double trisomy, 2n plus 1 plus 1, 2 added, plus 1 plus 1. Tetrasomy, this is the normal set, 2 sets are added, 2 individual sets of chromosomes are added. Pentasomy, 3. So, 2n plus 3. The condition is 2n plus 3 is pentasomy. These all are by addition of chromosomes, extra chromosomes within the normal set, normal diploid set. Hypoploidy. Hypoploidy, deletion of chromosome number. The chromosomes can be deleted. When it deleted, it is again classified into monosomy and nullisomy. There are 2 different types of hypoploidy. Hypoploidy is differentiated into 2 different types or classified into 2 different types, monosomy and nullisomy. Mono, you know, mono, single. Nullisomy. Nullisomy, it leads to lethal of an organism. So, it leads to death of the organism, lethal, nullisomy sometimes. So, hypoploidy, hypo, deletion of alteration by means of deleting the chromosome number. Within the normal diploid set, in monosomy, this normal condition, this is normal, normal set, one can be deleted. So, the condition is 2n minus 1. This condition is 2n minus 1. That is monosomy. And nullisomy, 2n minus 2. So, this is 2n. Sometimes double monosomy occurs. Double monosomy, it deleted 2n minus 1 minus 1. Double. Nullisomy, 2n minus 2. So, how it is? 2n minus 2. From the normal set, these two are deleted. So, it is 2n minus 2. This one. Within the normal set, these two are deleted. 2n minus 2. Here 2n minus 1. So, monosomy and nullisomy. Flowchart is important. First, you learn these names. Ploidy is classified into aneuploidy and euploidy. Aneuploidy. Now, these all examples or comes under aneuploidy. Within the diploid set, addition and deletion. So, it is classified into two types, hyperploidy, hypo. Hyper means addition of chromosome, hypo deletion of chromosome. So, it is classified into tri, tetra and pentasomy, hyperploidy. Hypoploidy is classified into monosomy and nullisomy. Now, euploidy. Euploidy is the changes or alterations observed within the entire set of organisms. The euploidy is classified into diploidy, haploid, diploid and polyploid. So, here it is known as haploid, diploid, polyploid. Haploid, diploid is the basic set which derived from paternal and maternal. Here is the homologous chromosome which is derived from maternal and another derived from paternal. So, this is a diploid set. Homologous chromosome 2n. Haploid. Haploid set. This is n. Haploid. So, haploid and diploid set of chromosomes. Euploidy, here it is classified into haploid, diploid, monoploid, diploid and polyploid. So, monoploid, for example, this chromosome to homologous chromosome, when it derives from paternal and maternal, it is a diploid set, homologous chromosome. The half number is called as haploid. So, diploid and haploid. Diploid and haploid. Another condition is there, monoploid. The monoploid occurs when the organisms under polyploidy. So, polyploidy means, poly means many. So, under euploidy, within the entire set of chromosomes, either addition or deletion, the chromosomes are altered. When the chromosomes are altered, monoploid, diploid and polyploid occurs. So, this monoploid, for example, there is a wheat, common wheat which contains 2n is equal to 6x is equal to 72. 6x is equal to 72. So, here diploid set, this is diploid 2n, and the haploid number of chromosome is n is equal to 36, half number of this, so 36. But this is monoploid. So, X is monoploid, X is 12. So, this is monoploid number. So, this monoploid is the condition when the organisms under polyploidy, this monoploid condition. So, this is haploid and diploid. So, haploid and diploid number generation after generation it is regular. Generation after generation the haploid and diploid number is regular. And this euploidy is classified into monoploid, diploid, polyploid. So, let us talk about this polyploidy. Polyploidy. Poly means more than 2 sets of chromosome. Addition of more than 2 sets of chromosome within the haploid set. So, this polyploidy is again classified into autopolyploidy, allopolyploidy. There are two different types of polyploidy, autopolyploidy and allopolyploidy. Allopolyploidy. There are two different types, autopolyploidy, allopolyploidy. The difference among these two are auto, within a same species. Addition of chromosome sets within a same species is called auto. Auto is self. Within the same species, if any alterations increased in number is called autopolyploidy. Allo with different species. Within different species, if any alterations can be observed is called allopolyploidy. So, this autopolyploidy is classified into triploidy, tetraploidy and so on. Triploidy, tetraploidy. Polyploidy, in plants, in animals it is rare, but in plants it can be induced, polyploid. To improve the varieties it can be induced. Triploidy, there are so many examples are there, cultivable banana, grapes, so many examples are under triploid condition, triploid. So, the chromosome numbers within the species can be altered, increased. Tetraploidy, there are some examples, wheat and grapes, tomato. So, these examples are tetraploidy. So, triploidy and tetraploidy, these are comes under autopolyploidy. Allopolyploidy, allopolyploidy within different species. Within two different species, there is a example of this allopolyploidy, Raphanobrassica. There is an Russian geneticist who crossed between two different species, allopolyploid, increasing the chromosome number of this. It can be induced by altering, doubling the chromosome number by means of colchicine treatment. So, here there is an example, Raphanobrassica, a cross between radish and cabbage. So, first example, there are two examples given in your book. This cross between a radish and cabbage. Radish and cabbage. The parent is radish, Raphanus sativus. Raphanus sativus. This is cabbage, Brassica oleracea. Brassica oleracea, cabbage. So when they are making a cross, so these two organisms possess the diploid set of chromosome 2n is equal to 18. 2n is equal to 18. This also 2n is equal to 18. Raphanus sativus and Brassica oleracea, the diploid set of chromosome is 18 in number, 18 and 18. So this is the parent. In the F1 generation, when they make a cross between these two, in F1 generation, it produces a sterile plant. So F1 generation, it produces a sterile, and this F1 sterile plant chromosomes are doubled by colchicine treatment. So next generation it becomes fertile. So this geneticist, Russian geneticist, they expected the edible plant, complete entire plant should be edible by giving the cabbage leaf part become a cabbage and the root part become radish, but the result they observed is vice versa. So they were disappointed, greatly disappointed. Then another example of this allopolyploidy, that is first man-made cereal. First man-made cereal. Let's see that the first man-made cereal is called as triticale. There are two different species, cross between two different species. That is, the first man-made cereal is called as triticale. A cross between Triticum durum and Secale cereal. Triticum durum is a wheat variety called Triticum durum is a wheat variety, and Secale cereal, this is a rye. A cross between, this is an example of allopolyploidy, increasing the chromosome number within different species. So altering by means of different, this is a wheat variety, wheat, and this is a rye. A wheat variety and a rye. So this Secale cereal, and this is a wheat and rye. The name of the wheat is Triticum durum, Secale cereal. So the chromosome number, these are selected as a parent. The parents are Triticum durum, a wheat variety, and a rye Secale cereal. The chromosome number is 2n is equal to, in this Triticum durum, in Triticum durum 2n is equal to 4x. X is 4 equal to 28. And Secale cereal 2n is equal to 2x is equal to 14. 2n is equal to 2x is equal to 14. This is the normal condition of this. In Triticum durum, first you know learn this number, diploid set of chromosome, 2n is equal to 4x is equal to 28. X comes under monoploid. So this is diploid, 2n is equal to 4x is equal to 28. Here 2n is equal to 2x is equal to 14 regarding this Secale cereal. And so when the parents are allowed to cross, this is a tetraploid, 4x. So 4x is tetraploidy, this is 2x diploidy, a cross between tetraploidy and diploid. So this is tetraploidy, this is diploidy. Now in the F1 generation, when there is a cross among these two in the F1 generation. The gametes, what are the gametes from here? The gametes are here, N is equal to, here gametes from triticum durum, N is equal to 2x is equal to, here half number. So, from this 28 it is 14. This is diploid set 2n. So, gamete, at the time of gamete formation, half number n is equal to 2x is equal to 14. Likewise, here at the time of gamete formation, here 2n become n is equal to, here x is equal to 7, half number. When you take out the gamete, here half number. So, this is the F1 generation. F1 hybrid, it produces n is equal to 2x is equal to 14 into this n is equal to x is equal to 7. So, when you make these two crosses, it produces, after gamete formation, it gives this n, here 2n is equal to n plus n, 2n is equal to 3x is equal to, here 21. 2n is equal to 3x is equal to 21. So, now it is triploid. So, this triploid, when they double the chromosome number, so this triploid can be doubled into hexaploid. So, when it is doubled, this 2n is equal to 6x hexaploid, it becomes double the chromosome number. So, this 21 become 42. So, here it obtains the name triticale, this triticale. By doubling the chromosome number, it produces triticale. This is the first man-made cereal. So, which is the first man-made cereal? Triticale. One word question, triticale is the first man-made cereal. So, this is very important. For 5 mark question, this can be asked. Allopolyploidy. There are two examples for allopolyploidy. First one, radish and cabbage, cross between radish and cabbage, that is Raphanus sativus and Brassica oleracea. Here another example, first man-made cereal. Most of the questions you find this. Very important, 5 mark also they may ask, or the man-made cereal is known as triticale. Cross between triticum durum. Triticum durum is a wheat and a rye, two different allopolyploidy, increasing chromosome number with two different species. Triticum durum and Seeker cereal, a wheat and a rye. Cross between a wheat and a rye is called this, and obtained a product is called triticale by doubling the chromosome number. How to doubling the chromosome number? By treating colchicine. When you apply colchicine, it doubles the chromosome number. So, here this 2n is equal to 4x. This is the tetraploidy, this is diploidy. These all are examples of polyploidy. Polyploidy, the number of chromosomes can be increased. Poly, many. More than 2 sets of chromosomes, 2 or 3 more sets of chromosomes can be increased. So, here it is induced by doubling the chromosome number by colchicine treatment. The triticum durum, common wheat, this one of the wheat variety, the 2n is equal to 4x is equal to 28, and Seeker cereal, the 2n is equal to 2x is equal to 14 in number. This is diploid, this is tetraploid. When make a cross between these two, the gametes which gives 14 and 7. So, half number, and when they double this F1 hybrid is doubled by means of Colchicine treatment. It produces this 3x, here 2x plus x 3x, n plus n 2n, x 2x plus x 3x. So, it gives 21. Now, this is sterile. So, when they double the chromosome number, this triploid becomes hexaploid 6x. So, this is an example of hexaploid 42. Now, it produces triticale. The name of this cereal is triticale, which is the first man-made cereal. What's the significance of this? Is there any significance of ploidy? Normally, if the chromosomes are altered, it's an aberration. Chromosome number or structure, if it is changed, it's impossible. In animals, it's very rare, but in plants, it gives more vigorous varieties. So, why it is plant breeders are using this colchicine treatment or by doubling the chromosome number to improve the crop. So, it is one of the breeding technique to improve the crop. Here it gives a more vigorous, and these plants autotetraploids and this autotetraploids, mainly this tetraploids 4x autotetraploids. Here it is again classified. Polyploidy is autopolyploidy, allopolyploidy. This autopolyploidy is classified into triploidy and tetraploidy. So, this tetraploidy is important for improving the ornamental plants. The ornamental plants can be developed. Ornamental plants are used to the ornamental. What are ornamental plants? The plants which are grown for beauty. So many plants, pteridophyte varieties or so many ornamental flowering plants can be altered the chromosome number by tetraploidy and produces more ornamental varieties, ornamental plants. And the height and weight of the seed and the height of the plant, higher weight of more water content, sweet content, these all increase by doubling the chromosome number. That means, these all techniques are involved by improving the variety qualitatively and quantitatively. So, it leads evolution of plants. In plants, it leads evolution, but if any lethal occurs, it causes lethal also, nullisomy, which causes death of the organism. But significance, this autopolyploidy, tetraploidy, triploidy, these all are by improving. One of the man-made cereal is developed, triticale. So, new varieties can be developed and it leads to evolution. So, just recall what you learned today. Chromosomal aberration. Chromosomal mutation is otherwise called as chromosomal aberrations. Alterations in number or structure. Structure, let us see in the next class. Today you concentrate this ploidy. Very important 5-mark question. Ploidy, the types of ploidy, various types. It is classified into this aneuploidy and euploidy. Then aneuploidy again classified into hyperploidy, hypoploidy. Then this hyperploidy is tetraploidy, triploidy, tetraploidy, pentaploidy. Here hypoploidy again classified into monosomy, nullisomy. Here triploid, tetraploid, pentaploid, and this polyploidy, here euploidy, it comes under polyploidy, monoploidy, diploidy, polyploidy, and classified into autopolyploidy, allopolyploidy. Here autopolyploidy classified into triploidy and tetraploidy, and allopolyploidy you learned 2 different examples, one from radish and cabbage another one triticale. So, learn all this question and draw this in the class note. Thank you.